Hello, my name is Sarah Copeland, and today I will be giving an introduction to mitochondrial DNA NGS data analysis using GeneMarker HTS software. GeneMarker HTS was developed in collaboration with leading laboratories and provides very sensitive detection of variants, including SNPs, indels, and heteroplasmy. The software has been validated for use on casework samples in forensic laboratories. GeneMarker HTS is forensic analysis software for data from NGS platforms such as the Illumina and Ion Torrent. Audit trail and user management are included with the software, along with comprehensive reporting options. GeneMarker HTS provides rapid analysis of NGS data for forensic applications such as database or casework samples, SDRs, YSTRs, mitochondrial DNA, or all three simultaneously. The mitochondrial DNA application allows for analyzing whole mitogenomes or specified regions such as hypervariable regions 1 and 2 or the control region, with multiple alignment options, forensic nomenclature, and the ability to upload profiles to MPOP. The STR application allows for analyzing autosomal and YSTRs using forensic nomenclature. Allele calls are concordant with CE results, but also include sequence variants to visualize isoalleles. In this webinar, I will be focusing on mitochondrial DNA analysis in GeneMarker HTS, but I encourage you to watch our webinar devoted to STR analysis as well. GeneMarker HTS offers multiple mitochondrial DNA alignment options, including alignment to the reference. The revised Cambridge reference sequence is the preloaded reference sequence, or you may opt to import a custom reference. In either instance, the alignment will span the origin. Consensus alignment can be used for improved detection of indels. And GeneMarker HTS also has a unique motif alignment, which provides recognition and proper assignment of SNPs and indels that is consistent with phylogenetic and forensic considerations. The program's motif alignment automates the motif recommendations of the DNA Commission of the International Society for Forensic Genetics and decreases analyst intervention. Traditionally, alignment is focused on minimizing the number of differences between the read and the reference. This is not always optimal for forensic analysis since there is an established convention for the positioning of many common variants. Each motif is a list of variant calls which are translated into an expected sequence. When using the motif alignment, the motif regions are designated by a hash symbol before the locus designation. Reads that cross the region will be adjusted to match one of the possible variants in the list, while reads that either start or end in the motif regions will be soft-clipped. As you can see in the table, the traditional alignment has 2 deletions and an insertion, while the forensic alignment has 4 substitutions and 1 deletion, which matches the motif shown in the green box below. Motifs can be easily updated in the motif editor. GeneMarker HTS has a wide range of reporting options: consensus sequence, variant reports of SNPs, indels, and heteroplasmy, comprehensive coverage, easy import of results into MPOP for haplotype information. GeneMarker HTS is easy to use in a Windows interface and allows for the user to customize analysis parameters, filters, and reports. Before we start a new analysis, there are some setting options that can be accessed on the main screen. Using the gear button on the upper left of the screen, the user may customize colors for the display, change the license type, optimize performance for the software, log into the server, and enable user management. The upper right corner has the About button, listing information about the version of the software, and the Help button will open a linked user manual. The center has options to start a new analysis, open a previously saved project, or manage settings such as panels and motifs. Now let's start a new analysis. The Project folder field allows you to enter a location to save all of the reports. If nothing is entered, the program will save the reports to the folder the data files came from. The default reference path is the revised Cambridge reference sequence, but you can also import a custom reference file. There are default motifs, but you can add your own by deselecting Use Default Motif and going to Create Motif. Here you can add or delete individual motifs or entire regions. If you are using one of Promega's PowerSeq kits, like the Promega PowerSeq CRM or Promega PowerSeq WGM, you can choose to use the corresponding panel. However, for this webinar, we are going to use shotgun sequencing data, so I will select None. The panel method for mitochondrial DNA analysis is the topic of a separate webinar, which we encourage you to watch. There are various alignment options and filter settings that you can customize, as well as the ability to mask personal health information. Sample files can be added by using the Add icon in the lower left-hand corner. I'm going to analyze whole mitochondrial DNA paired-end data from a MiSeq to demonstrate the workflow. When loading paired-end data, the files will be grouped accordingly. Samples can be removed individually or all at once using the Remove or Remove All buttons. Now we can press OK. This pop-up is just stating that a folder will be created for the reports since I didn't designate a location for it. As samples finish, they will become available in the Viewer window. You can open up and view a sample while the others are being aligned. This is the Alignment Viewer screen. At the top is the Global Viewer, which displays the depth of coverage with forward reads in blue and reverse reads in red. Variants are denoted by green vertical lines. Below is the reference sequence, in this case the revised Cambridge reference sequence, and the consensus sequence. In the center are green blocks indicating the location of motif regions, and if amplicon regions are used, there will be blue blocks indicating their locations as well. Below is the pileup of reads, including arrows indicating the forward and reverse reads. To sync the global viewer with the pileup, select Zoom Global. To zoom in, draw a box from left to right. Scroll by holding down the right mouse key and dragging. Zoom out by drawing a box from right to left. The very bottom is the variant table, which gives the user the option to view variants for the major profile, minor profile, or both. Now that other samples have finished, we can open multiple viewers at once to enable synchronized viewing. The Comparison Viewer provides sample-to-sample comparison of major and minor variants, as well as a percent variant comparison table. Results are saved automatically when the sample is analyzed. Any edits to existing projects must be saved as new reports. Doing so will also save the edits as part of the audit trail in GeneMarker HTS software. Result tables for each sample include BAM files, consensus sequence, major variant, minor variant, and alignment statistics log. The major report has the format needed to copy-paste the variant list into the MPOP database for haplogroup information. Thank you for your interest in GeneMarker HTS software. For more information or for a free 35-day trial of GeneMarker HTS and other SoftGenetics products, please visit www.softgenetics.com or email info@softgenetics.com. For technical support questions, please write to tech_support@softgenetics.com. Thank you for watching.